In this section, I'm just going to give you an overview of a few of the protozoan pathogens, some of the more common ones. So protozoan identification and cultivation. So how do we know that a person is infected with a protozoan? Most commonly, it's going to be in their stool. So you take a stool sample, you spin it down, you have to go through this fecal concentration technique because raw stool, if you just look at it, everything in there looks like a protozoan. All the undigested food matter and plant matter and all sorts of stuff can look like these protozoans. So if you do a concentration technique, you're trying to concentrate the stool down to its parasitic essence. So only the parasites are left in the stool, or the parasites are concentrated in a stool sample. In order to investigate stool and to say for sure that the patient does not have a protozoan infection, you actually have to collect 3 stool samples on 3 separate days and confirm negative results on 3 slides for concentrated stool specimens. So you really have to look hard before you can say, nope, they don't have a protozoan infection. So considerations in identification, the shape and size of the cell. So this organism, the single-celled organism, a lot of them look alike, and I'll show you some pictures of them, how close they look. So having a micrometer in the microscope and actually measuring the size of the sister trophozoite can help you identify what pathogen is causing the infection. When we're treating this, we're basically treating them with the same drugs. So we're only doing identification for source tracing. If we know that someone's coming in with a parasitic infection, we want to know what type of parasite it was. And then if we have multiple cases coming in with that same parasite, we can kind of say, okay, where did all of these people just eat? And then we can go track it back to its source and prevent other people from getting sick. So it's really the predominant reason why we need identification, is so that we can report to the state, yes, we made sure that this is the species and this is the only reported case, or yes, this is the species and we had like 50,000 other people come in with this. We need to figure out where this is coming from to stop the spread. So again, size and shape of the cell, type, number, and distribution of the locomotor structures, the presence of specialized organelles or cysts, and the number of nuclei. Blood, sputum, which is just deep spit, cerebrospinal fluid, fecal or vaginal specimens smeared on a slide with or without staining are the most common specimens that we would collect. So parasitology is the study of protozoa and helminths, and a parasite is the term that's most often used to denote a protozoan and helminth pathogen. There's a bunch of different types of pathogens, and here's a huge list of some pathogens that you can take a look at. So amoeboid ones, Entamoeba histolytica, Naegleria, Acanthamoeba, and what they cause. Ciliated would be Balantidium coli. Flagellated include Giardia lamlia, Trichomonas vaginalis, Trypanosoma brucei, and cruzi. Leishmania, a bunch of different types of Leishmania exist. And then the non-motile apicomplexans include Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, Nawalsi, and there's a bunch of other Plasmodium species that cause malaria. Toxoplasma Gondii, Crypto, and Cyclospora are also pretty common. So in this course, you're responsible for knowing the ones that I go through in detail. So starting now through the end of this segment, you should be familiar with the different types of parasites that I'm going to go through. So the first one is Giardia lamlia. Giardia lamlia is easily identifiable in its trophozoite stage because it kind of looks like a smiley face. So you can see the 2 nuclei, and then it kind of looks like a little smiley face there, and you can see the flagella hanging down that kind of look like hair and arms and legs and so forth. So it kind of looks like, haha, I'm about to give you diarrhea. This one will be transmitted by drinking or eating cysts in fecally contaminated water or food. The mode of action is it coats the surface of the intestines to prevent nutrient absorption. And this one causes symptoms of diarrhea and greasy stool because it's not allowing for the absorption of nutrients from the digestive tract. Prevention and treatment, there is an animal vaccine that exists. There isn't a vaccine for humans yet, but this is easily treated with drugs. Giardia is one of the most common protozoan diarrheal infections out there. The way to distinguish this one from other ones, with the exception of looking at it, is the fact that it coats the surface of the intestines. So that's how you know that it's Giardia. If it's coating the surface of the intestines, it's Giardia. Trichomonas vaginalis is also a flagellate. Trichomonas, and actually, let me just correct the spelling here real quick. All right, much better. Okay, it's also a flagellate. So we have all of these flagella, this tuft of anterior flagella, and then there's a posterior flagella too. And then there's this undulating membrane that helps it move. These organisms are pretty large, and they can colonize the vagina. They cause a foul odor, itchiness and irritation, a white-gray or yellow-green discharge, and painful sex. If you have Trichomonas, you definitely know that you're infected. So Trichomonas vaginalis, it's a very uncomfortable sensation. You have these large protozoan organisms. When you look at this under a microscope, typically you go in with these symptoms. They will try to look at it microscopically with just a vaginal wet mount. They're going to take a swab of fluid from the vagina. They dip it in some solution, so typically like a saline type solution, smear it on the slide, and then do direct microscopy in a wet mount on the slide. You can see these organisms, and sometimes you can see them moving around. They spin around in circles when you look at them under the microscope, and they're huge. In the background, that's some bacteria that's stained, so you can see how much bigger than bacteria these things are. This is the highest #1 cause of STDs worldwide. So Trichomonas is the most common STD in the world. Trypanosoma cruzi. So Trypanosoma cruzi is also a flagellate, and this is what Trypanosoma cruzi looks like. So that's the organism, and this is what transmits it. It's transmitted by the stool of the kissing bug, which enters into the blood. So this little organism is inside of this bug's digestive tract. These bugs are found in southern United States, Central and South America, and they carry this Trypanosoma cruzi, which causes Chagas disease. There is a similar version of this called Trypanosoma brucei, which causes African sleeping sickness over in Africa. But in this class, we're just going to focus on Trypanosoma cruzi and Chagas disease because it is in the United States. So this kissing bug will climb on you while you're sleeping, and then it usually bites around the mouth, and that's why they call it the kissing bug, because this triatomy-like insect is going to bite you around the mouth. It can bite in other locations. It's just pretty common to bite around the mouth. In southern United States, I used to live in Texas, these things were all over the outside of my house. We don't know if they're infected or not, but I know that these bugs were everywhere, and they were biting my dogs that I kept outside and everything. So this is a pretty common insect to see down there. So when this thing bites you, it's going to suck your blood. And when it eats, it has to poop. So it makes room inside of its abdomen by pooping. It'll defecate, and then either you're going to scratch it because it's really itchy when they bite you, and you're going to scratch the stool of the kissing bug into your bloodstream, or it'll scrape its butt along your skin and directly insert its stool into the open wound. So we don't know why this insect's doing that behavior, maybe to try to cover its tracks or cover up the blood spot, but that's just one of the behaviors that it does. So there's a couple different ways that this can get into your body. Once in the bloodstream, it's going to travel to your muscles, and it's going to replicate in muscle cells and macrophages and then spread to other organs. Symptoms include inflammation, fever, which can progress to heart and brain damage, which can be permanent. Prevention and treatment: bug spray. Acute cases are treated with drugs, but chronic cases can be fatal. So acute means short-term, rapid onset. Chronic means that you've had it for a long time and it's not going away. So chronic cases can be fatal. Prevention is also through bug nets, and in certain parts of the world, they have roofs with thatch houses with thatch roofs. So not having that type of building structure, having solid walls in your house to prevent bugs from entering into your house, that's also a way to try to prevent Chagas disease. And I don't see Chagas on the slide here, so I'm just going to write it down so you have the correct spelling. Chagas disease. Entamoeba histolytica. Well, this is an amoeba. Transmission is by swallowing cysts in contaminated water. The mode of action, the cysts are going to hatch open in your intestines, and then the trophozoites are going to start to digest your intestinal wall. Entamoeba histolytica causes dysentery, diarrhea, so severe abdominal cramping and pain, diarrhea, fever, weight loss, eventually hemorrhage and perforation, leading to the spread of the disease to other organs. So what this one does is, yeah, it causes diarrhea, but if it sticks around in the body long enough, it can actually eat holes through your intestine, and it can cause your intestine to perforate. It can also cause small amount of bleeding in your intestine and internal bleeding, which can lead to anemia. If these perforate through your intestines, they can spread to other organs and start eating other organs in your body. Prevention and treatment, you want to drink purified water, and this is treated with drugs. And then this is a picture of the trophozoite. So this is that picture that I referenced earlier. So you can see the trophozoite stage and the cyst stage of different species of amoeba. And you can see how similar they are. Some of them are larger, some of them are smaller. But if you were to get a stool sample and just have trophozoites in them, it would be pretty tough to distinguish. And this is only a fraction of those. So how we diagnose in a laboratory is we usually look at these under the microscope with the micrometer to help measure, and then we're going to investigate if we can see any cysts. Cysts are a little easier to distinguish. You can start to see these bodies inside of them. You can start to count the number of nuclei. We always know that Entamoeba coli has 8+ nuclei that you can visualize. And there's slight distinguishing features that you can notice inside of these. But as the trophozoite stage, it's largely dependent on size. Naegleria fowleri is an amoeba. Transmission of this one is entry into the nose while swimming in infected natural warm water. The mode of action, it penetrates the nasal cavity and enters into the brain. It will start encysting in your brain, and it can form these large cysts which destroy brain matter, and that causes hemorrhage, coma, and death. A very high percentage of people that get Naegleria fowleri die from Naegleria fowleri. There is a treatment for it. You can get treated with using those drugs, but usually once the symptoms set in, it's too late. And the people that do survive have ongoing, permanent disabilities, and they have permanent brain damage from this. So if you swim in natural bodies of water, predominantly in the southern United States and Central and South America, sometimes in the central United States, I heard of cases in Tennessee and Virginia and so forth. If you swim outside in natural bodies of water and the water is infected with Naegleria fowleri, it can splash up into your nose, and they stick to the cilia inside of your nose, and they can eat holes like straight through up into your brain. And that's where they get into your brain, and this is known as the brain-eating amoeba. Once they get in the brain, that's when they cause their major symptoms. This is pretty common. Cases are all over the United States. You hear about them all the time, the brain-eating amoeba. And instead of calling it the common term brain-eating amoeba, it's known as amoebic meningoencephalitis. Amoebic, meaning that it's caused by an amoeba. Meningo is talking about the meninges that surround the brain and spinal cord. Encephal is talking about the brain, and itis is inflammation. So it's inflammation of the meninges and brain that's caused by an amoeba. Balantidium coli is a ciliated one, and you can see the cilia all the way around the surface of Balantidium coli, and then you can predominantly see it on this surface in this picture. Balantidium coli causes diarrhea-like symptoms. It's treated with a protozoan drug, anti-protozoan drug, and this one is one of the only ciliates that can cause human infection. So then moving on to the apicomplexans, Plasmodium falciparum. So I'm going to talk about Plasmodium falciparum. It's the most deadly form of Plasmodium that causes malaria. There's other forms. Plasmodium falciparum is pretty common in Africa. Plasmodium vivax is more common in like Central and South America. Nawalsi and Ovali are going to be more in Malaysia and India and so forth, but all of these are found all over the place. This is one of the deadliest versions of malaria, so I'm going to talk about this one. So Plasmodium species cause malaria. The mode of action is it invades red blood cells. It's going to cause flu-like symptoms, cardiac arrest, and death. So what this one does, and you can see this picture of a blood smear, you can see this little oval ring inside of here, and that's how you can identify that this patient has malaria. It's a ring-like structure, and it looks like it has a little diamond on the top of the ring. With Plasmodium falciparum, usually you're going to have cells that have multiple ring forms inside of them. So the purple ones are white blood cells, the pale ones are going to be red blood cells, and then you can see the ring inside of the pale red blood cell. You're also going to see, oops, sorry about that. You're also going to see a large number of eosinophils. So if you remember, eosinophil is associated with parasitic infections. You're going to see more eosinophils because this patient is infected with a parasite. So what this thing does is a mosquito, an Anopheles mosquito that is infected with Plasmodium falciparum will land on you and bite you. That mosquito is a female because only females need to feed off of blood. Females are going to be laying the eggs and they need blood proteins in order to support the growth of those eggs. So female Anopheles mosquito lands on you, she's going to suck your blood, and to prevent your blood from clotting, she actually spits into you an anticoagulant. While she's spitting that into you, sometimes Plasmodium falciparum will come with it. And this isn't found in the United States to our knowledge today. It could come back to the United States, but this is predominantly in other parts of the world and in tropical countries. So when this parasite gets into your blood, the parasite is going to go to the liver. And when it gets in the liver, it's going to go through a couple different stages. When it finally emerges the liver cells, it's going to then get into your bloodstream. From the bloodstream, it's going to enter into your blood cells, and that causes your blood cells to become really sticky. And the blood cells are going to slow down as they go through your blood vessels, and they're going to stick to your blood vessel walls. So now you have sticky blood cells that are sticking to the inside of your blood vessels. Think about trying to go through a hallway and having all of a sudden you're stuck to the wall. You can't move through. So now you've got clogging up of inside of the blood vessels. And the reason why it's doing this is because when Plasmodium replicates inside of the red blood cells, those red blood cells look large and weird, and your spleen will kill them. So if those red blood cells make it to your spleen, it'll clear the infection. But the parasite is smart. It doesn't want those red blood cells getting to your spleen because it wants to be able to infect you. So it's going to cause your red blood cells to stick right where they are, right on that blood vessel wall before it gets to the spleen, and that's where it's going to replicate. Now all of these will infect in a cyclic manner. So you'll have maybe 100 parasites infect 100 red blood cells, and those 100 red blood cells will stick to different blood vessels in your body. And then all at once, kind of like there's a signal that says, hey, let's get out of here, they all burst open and rupture out of your red blood cells. When that happens, the patient has convulsions, an extremely high fever, and very severe symptoms. And those convulsions and high fevers will last for a few hours, and then the parasites will find new red blood cells to go into, and they'll infect those new red blood cells, and the symptoms go away. So then you're like, oh, that wasn't that bad, I feel better. Well, another day later, the same thing happens. Or another couple hours later, the same thing happens. So you have this cyclical eruption of these parasites that causes these massive symptoms all over your body. You have the flu-like symptoms, and then cardiac arrest and death can happen when these block your blood vessels. It's especially common when this gets into your brain. It can cause death that way, the brain version of malaria. And this can also happen in children and pregnant women, because children's blood vessels are so tiny, so these can get clogged up in the blood vessels and cause cardiac arrest. And pregnant women are managing much more large blood volumes, so they usually have more severe cases and are linked to a higher risk of death. The good thing about babies is babies can't get this up to about 6 months of age. Babies are born with fetal hemoglobin, and malaria parasite does not like fetal hemoglobin. So infants through about 6 months of age can't get infected. After 6 months to about 5 years of age, most high risk category. So to prevent this, they use bug spray. Mosquito nets are very common. There's certain seasons usually that malaria is a little bit worse, so you take more precautions during those seasons. If you're a tourist traveling to an area that's endemic to malaria, you're going to take prophylactic drugs. So usually you're prescribed with doxycycline or some other type of anti-parasite type prophylactic drugs so that you don't end up getting sick. And you're going to wear bug spray and take the necessary precautions. We used to have DDT around. So the United States had malaria. We came out with DDT and bug spray. We sprayed it all over the place, killed off the Anopheles mosquito, and eradicated malaria from the United States. DDT was an issue with birds, however. So predatory birds like hawks and eagles and such were susceptible to poisoning from DDT. And what happened with them is if they inhaled DDT, they would lay eggs with softer shells. So it would deteriorate the shell of their egg. So when they go to have babies, they're going to lay their eggs in their nest. They fly away, they come back to the nest, and because those eggs are so fragile, they would sit on them and crack them open. And our bird populations were just declining like crazy, which was linked back to this DDT, which was rapidly taken off the market. Because DDT is removed, there is a slight risk that malaria is going to come back to the United States. And this is treated with antimalarial pills. There's different types of medications, which we'll get into when we talk about antibiotics. These medications, there's a lot of resistance that's been developed. The parasite resists the medication, and they're really no longer effective. Long-term drug therapy is necessary, and usually you have to use 2 drugs. You can't just use 1 drug at a time. You have to try to attack it with 2 drugs at a time. Cryptosporidium parvum. If you were around in the 1990s in Milwaukee, in kind of the southern part of Milwaukee County, then you may have experienced the biggest outbreak of Cryptosporidium ever. So when I was little, growing up in Milwaukee, we all of a sudden had this huge outbreak, and I wasn't allowed to drink from the water fountain at school anymore, and we had to boil all our water. I remember my dad and my brother both ended up getting sick. Hundreds and hundreds of people ended up getting sick and dying actually from crypto. So transmission is consumption of infected water. The mode of action is it actually infects the cells of the intestine. So we talked about some of these that cause diarrhea. So Giardia coats the intestines. Entamoeba histolytica eats through your intestines. And now Cryptosporidium parvum infects the cells of your intestines. So it gets inside of your intestinal cells. Symptoms include severe diarrhea, headache, sweating, vomiting, abdominal cramps. They're treated with antidiarrheal drugs, usually not anti-protozoan drugs, antidiarrheals. The protozoan is easily attacked by your immune system and does not persist in a relatively healthy human host. So usually this is self-limiting or self-limited, meaning that it goes away all by itself within a couple days. If the patient is immunocompromised, it can cause death, and many of the people in the Milwaukee outbreak ended up dying of Cryptosporidium. Luckily, my dad and my brother were okay. They just were sick for a couple days and they recovered. This actually stemmed from the Milwaukee treatment plant. When you do testing, like water testing, the results that come up are really from yesterday's water. So it takes a day to get the results back. So by the time you notice that there's an issue in the water, it's usually too late. Crypto is not easily treated with the typical chemicals and doesn't die in the presence of chlorine. It thrives in the presence of UV light. So all of those factors make it a really infectious particle. So with crypto, you actually need an ozone generator to get rid of it. And then they've genetically engineered certain bacteria to be able to eat crypto. But the reason that it persists so long is because it doesn't die in the presence of chlorine. So if someone has crypto and they go into, let's say, a swimming pool at a busy water park or something like that, those spores can be left behind in the water. And because chlorine doesn't kill them, everybody that comes in contact with that water is vulnerable to getting crypto from that water park. So it's really difficult to stay on top of this and to make sure that we don't transmit this in the water, and ozone generator is really the only way to clear this from an infected water source. And now we're going to talk about Toxoplasma Gondii, which causes toxoplasmosis. So if you've ever heard of pregnant women not being able to clean the litter box, it's because of this one. So Toxoplasma Gondii is an apicomplexan. Transmission is by eating undercooked contaminated meat, inhaling cysts from, or oocysts, which are the eggs from cat litter. So with this one, the cat consumes meat that's infected. So the organism itself is within the muscles of mice and birds and a variety of other animals. So if it's a cat that goes outside, the cat's going to eat some mouse muscle and the cat is going to become infected. Now if the cat comes back into your house after being infected, the cat is going to pass those cysts out in their stool. Now you're going to come along and you're going to clean the litter box out, and those are going to aerosolize, and then you breathe them in. And when you breathe them in, they're going to hatch open and migrate to your muscle tissue, and they're going to stay in your muscle tissue forever, and there's not really a big issue with having them in your muscle tissue. They estimate that 90% of cat owners do have this in their muscle tissue, or up to 90% of cat owners have this in their muscle tissue. Now the only way for that parasite to complete its life cycle would be if a cat were to eat your muscle tissue. So since many of us don't get eaten by lions or tigers, usually we're considered a dead-end host or a terminal host. So it gets into our muscle tissue and it stays there, and it's not really a big deal. It is a big deal, however, if you're pregnant. If you're pregnant and this is the first time that you've ever been exposed, those cysts can get into your tissues of your unborn baby, and it can cause major birth defects in the baby, deafness, and even miscarriage. So it's something that we want to avoid. So symptoms, like I said, none. 90% of certain human populations are or have been infected at some time. Issue is in immunocompromised and AIDS patients or first-time exposure in pregnant women. In immunocompromised and AIDS patients, there can be pretty large inflammatory reactions, which can cause muscle pain and weakness. Treatment would be drugs. You only take the drugs if you're having active symptoms. Unfortunately for pregnant women, the drugs harm the baby. So if you get this and you're pregnant, there's going to be harm to the baby. So it's better to just stay away from a litter box. If it's your, if you have a cat that goes outside, or if there's any chance that this cat could be infected with this, then you want to stay away from the litter box if you're pregnant.